Mimicry is a classic example of the natural selection model. Species that are vulnerable to predators develop the same appearance, also known as phenotypes, as a protective device to imitate another species avoided by its natural predator. If a predator learns to avoid one of those noxious species, it will avoid the mimic species as well. However, this is just one of the many forms of mimicry seen in the wild, known as Batesian mimicry, with Mullerian mimicry having two toxic species and them converging on a shared warning signal, like stripes on honeybees and yellowjackets. Also, there is aggressive mimicry, being used by predators imitating its prey's food to hunt and nearly doubling the predator's effectiveness. In this video, I will discuss why mimicry is seen as a great source of fitness by species, what processes cause the adaptation, and some common results of mimicry, like speciation or background selection. Mimicry is everywhere in nature, from butterflies to snapping turtles to anglerfish. Prey species in particular have extremely strong selection towards being phenotypically similar to other species that are either detrimental or non-beneficial to their natural predators. The idea that all types of species could employ their own independent defenses or go through convergent evolution towards the same defense is possible, but as evidenced by nature, the individuals who look most like a species who already have a defense against a predator will be heavily selected for and have the higher fitness. For an example, if a small yellow fish was toxic to a shark, and a species of medium brownish fish was completely safe and beneficial to the shark to eat, they would begin to be under selective pressure. The smaller and more yellow-tinted fish out of the original medium brown fish population would begin to be heavily favored, as the shark already knows to avoid these fish. In Mullerian mimicry, it is stated that predators only need one negative encounter to avoid all future correlation, even if they were different species. Other types of mimicry, like aggressive mimics, are utilized by predators to trick their prey and can double their effectiveness at hunting. Some hawks travel with and appear similar to vultures, and snappy turtles use their jaws to appear like a worm to lure in prey by appearing like their prey. So what exactly are the forces that drive the positive selection? Both genotype changes and phenotypic plasticity are present in these mimic species. Mimic species will heavily favor traits that give off a similar phenotype to the model species that they are trying to imitate. This can be seen as a type of convergent evolution, as the phenotypes become so similar due to the model species being more fit than the mimic species against a predator. The phenotypes of most species that incorporate mimicry are polygenic traits with multiple autosomal loci, which can lead to epistasis. Epistasis is present as the mimics are selected as the genes that cause canalization towards phenotypes. An example of this could be how less defined patterns in species of butterfly move towards the model species phenotype. In the opposite direction of genotypic changes in mimics, phenotypic plasticity is also prevalent in mimic species. Phenotypic plasticity allows mimics to adjust their appearance, behavior, or other traits in response to environmental conditions. This flexibility can enhance mimicry accurately by enabling mimics to closely match the characteristics of their models under varying environmental contexts. It has also been seen to support selection for transition from Batesian to Mullerian mimicry. Finally, mimicry in an ecosystem can have some notable effects, mainly a fast tempo of speciation, with mimicry being the mode of said speciation. As seen in the study of H. cydno and H. Melpomene butterflies by Russell E. Nasbitt, Christy Jiggins, and James Mallet at the Galton Laboratory of University College London, they found these two phenotypically different butterflies are actually two sister species that recently diverged in order to mimic two completely different species of Heliconius. Due to the loci that impact the color and patterns on the butterfly being so closely linked together in two supergenes, leads to the linkage of said genes. This causes a barrier to gene flow between these mimic species, as their hybrids are not able to mimic either of the model species, even though they were recently the same species. Overall, mimicry is an astounding testament to the ingenuity and complexity of evolution as a whole, even though it is just one form of natural selection at work. It gives us insights into many aspects of evolution, like speciation, adaptation, can even be used to see differences in genotypes and phenotypes between species' converging traits.